Okay, so let's continue with yeast assembly, which is also known as gap repair. So usually, and this is just to let you know what kind of like what the search terms are when you want to use that method. So gap repair is usually used if you clone a single gene into a plasmid, and yeast assembly is used if you kind of like clone many genes into a plasmid. But of course, there's no patent on these terms, but they're usually interchangeable. Okay, so let me move this away. Yeah, so as we talk about DNA assembly, we do not only, or we cannot, like a way of not doing it with enzymes is actually using yeast for in vivo assembly. And this is possible through its endogenous homologous recombination repair mechanism. So this is basically a way how you can assemble parts of fragments that we just talked about, not using enzymes, but rather using kind of like the endogenous assembly mechanism of an organism. And I'm just gonna briefly jump into the mechanism behind that. So homologous recombination is basically the dominant repair mechanism that Saccharomyces cerevisiae, and this is when I talk about yeast, I usually talk about Saccharomyces cerevisiae, uses to repair any kind of DNA damage. So any organism needs to have mechanisms to repair DNA damage. And many mammalian organisms, including humans, actually use something that is called non-homologous end joining for this, where basically kind of like a nick in the double-stranded DNA part is joined again together by ligation. But Saccharomyces cerevisiae actually, interestingly, predominantly uses homologous recombination as its repair mechanism. That means that the nicked ends actually look for homologous region and then through kind of like strand invasion and synthesis, repair this by homologous recombination. And this is of course here depicted in the context of a genomic DNA recombination event. But we can harness that in the laboratory in order to actually join pieces of double-stranded DNA as long as they have homologous regions encoded at their end. So bottom line, everything that we can do with Gibson assembly, we can also do in yeast just through its endogenous enzymes that are present. To go back, so this is also why we kind of like lumped it together in this webinar, because Gibson assembly and yeast assembly are basically can be basically used interchangeable as the same genetic parts can be used for it. This means namely fragments with homologous ends of, let's say, 40 base pairs. So Gibson assembly usually works between like 20 and 40 base pairs, and yeast assembly you want to have a little bit longer ends, but it usually works between like 25 and 40 base pair homologous regions. So this makes yeast assembly actually very cheap because you don't need to buy any enzymes. But yeast also grows slower than E. coli. So the whole process from actually generating your fragments to an assembled gene or pathway takes about 2 to 3 days. But this is only incubation time. So that means you can't do anything else in between, but just be aware there's a little bit more time involved. So really to kind of like compare that in a graph. So we use the same fragments, a linearized plasmid, and I'm just going to use the same example as Ian did before, our DCP1 gene and our RFP fusion to it. Gibson assembly, you basically mix the fragments together, you mix the enzymes. This, let's say, takes you an hour. Then you transform it into E. coli, and after overnight incubation, you get colonies, which hopefully have your assembled plasmid. With yeast assembly, you use the same fragments, you mix them all together with the yeast strain, and I'm going to go into a little bit more detail how that reaction is going to be set up. And then you transform that, and after 2 to 3 days, you get colonies on your plate. And then again, you need to confirm that you have the assembled plasmid. The one difference you need to keep in mind is here, you really need to work with a yeast cloning vector. Of course, E. coli and Saccharomyces cerevisiae are 2 different organisms, so they need different components in the plasmids for replication. So we need a yeast origin of replication and also yeast selection markers present on these plasmids. And again, I will in the next slides go a little bit deeper, but keep that in mind. It's not exactly the same components, but the genes gonna stay the same, and also kind of like the concept of these homologous ends are the same. So when does it actually make sense to use yeast assembly over Gibson assembly? So first of all, then you work with yeast as your final cloning host. Of course, then yeast assembly should be your go-to cloning method, because eventually you anyways want to have your plasmid in yeast, and it doesn't make a lot of sense to go via Gibson assembly. But there's also, even if you work, for example, with E. coli, there is certain instances when it makes sense to consider yeast assembly as the method of choice. And this is when you assemble many fragments. So we already kind of like discussed that in the Q&A session. When you have more than 3 pieces, and this is kind of like an example that I took from the iGEM 2012 Frankfurt team that kind of introduced yeast assembly as a cloning standard. So here they kind of cloned 8 pieces together, like 3 different genes and always a terminator and a promoter. And this is usually very easily achievable in yeast assembly, but will be difficult for Gibson assembly. The other instant is when one or more of the fragments are really short. So for example, you have a promoter in here, which is only 50 base pairs long. So as Gibson assembly actually has an enzymatic reaction, the T5 exonuclease reaction, that chews back the 5' ends of your DNA parts. If the DNA parts are really short, you run risk that they're like chewed away, and often that introduces mutations. So it doesn't mean that Gibson assembly doesn't work at all with very short parts, but often you need to screen many colonies to actually find ones which do not have mutations. With yeast assembly, this is not an issue because it doesn't rely on actually like chewing back double-stranded DNA. So these are really the 2 cases when you should consider yeast assembly as your method of choice for DNA assembly. And then after assembly in yeast, given your actual host organism is not yeast, you can subclone your gene circuit into any user-defined vector. So for example, you did your assembly of your 8 pieces in yeast, you can then design primers which bind at the end, and then, for example, use Gibson assembly to just clone that or subclone that into another vector, and then you only have a 2-piece assembly. And this might seem complicated, but it is actually often the way to go to assemble large circuits. Just to kind of like give you an example of the usefulness of yeast homologous recombination in science. So for example, the whole mycoplasma genome was assembled from 25 overlapping DNA fragments in yeast in 2008. Actually afterwards, only afterwards, Gibson assembly was invented, but the first method of choice was yeast assembly. And then also currently in 2018, a full synthetic chromosome, actually several full synthetic chromosomes of yeast were assembled in vivo, not by a different, like technically a little bit different mechanism than classical yeast assembly in a plasmid, but still harnessing the homologous recombination mechanism of yeast. So it's really powerful, reliable tool to do in vivo assembly. Yeah, so let's see more technically yeast assembly step by step. So what you need is basically 3 components. You need a yeast cloning strain, and you can ask any of your colleagues for any strain that is out there. Maybe you have someone in your department that works with yeast. Most importantly, it needs to have auxotrophic markers, because then in yeast we usually don't work with antibiotic markers, but rather with auxotrophic markers. That means single genes of the biosynthetic pathways of several amino acids and bases are deleted, such that you can bring them back in the plasmid and then select for the ability to grow in a media that lacks these amino acids. So a common strain that is available is that is used by many people is the BY4741 strain. And I gave you the number from ATCC here, where you could order it in case you don't have that in your lab available. And the second component you need is a yeast plasmid or a yeast backbone. And here I recommend the PRS series, which like nicely complements all these auxotrophic markers. So these strains and the PRS series actually work very nicely together. And the PRS series is really like a modular Saccharomyces cerevisiae E. coli shuttle vector system where you have all the components to replicate in E. coli and in Saccharomyces cerevisiae. And again, I added some of the add gene numbers where you can get several different of these modular vectors to order. And then the third component you need is your PCR, your amplified PCR fragments. And Ian has told you before exactly how that works. So you can really use the ones you already maybe designed for your Gibson assembly, or just, you know, use any tool in benchling that is available for Gibson assembly. You can use that as a design principle to also design your yeast assembly primers. Again, keep in mind your linearized plasmid needs to be a yeast compatible plasmid. And then in general, 25 to 40 base pair homologous overhangs. So this is the three pieces, or like the three components you need. And then you can basically get started. So again, you have generated your fragments by PCR, and the yeast transformation basically works by a so-called lithium acetate PEG method, which I have posted in my resources slide on the last slide. So you're going to find a method how to do that. It's very easy. It's very similar to making chemically competent E. coli cells. And all you need to do is kind of like mix the yeast cells, mix the component of this method, and then 100 nanograms of your plasmid amplified, and then 4 to 10 molar ratios of each insert. You all mix that together, and then you follow the protocol, and you plate your cells, and you select for colonies on the appropriate selective media, depending on the shuttle vector you're using. And after 2 to 3 days, you have colonies growing on your plate. And usually many of them have the correct insert, but still similar to Gibson assembly, you still need to verify that the assembly actually works. And you can do that by colony PCR, by designing primers that, for example, or you can even use the same primers that, you know, this guy that binds at the end of it and this at the beginning, to see if they have been assembled into one piece. And then you can either send this colony PCR directly after purification to Sanger sequencing, or you can extract plasmids from yeast and then bring them back into E. coli. So usually plasmid extractions from yeast don't give very good Sanger sequencing results. You don't get high concentration, and usually they're contaminated with protein. So I recommend to retransform them into E. coli, do a mini prep, and then send them for Sanger sequencing. But again, usually let's say 2, 3, 4, 5 piece assembly works very reliable. So if you pick 10 colonies, for sure you're gonna have, you know, at least 3 or 4 with the right assembly. Yeah, and with this, I'm already through with my presentation. So as resources, I posted for you actually the website of the iGEM team Frankfurt 2012, because they really put together some nice resources on what to consider for yeast assembly or gut repair. They have some protocols out there, and they kind of like try to establish that as a cloning strategy for iGEM. So it's worthwhile to have a look. Then I posted here a benchling protocol for actually doing this lithium acetate polyethylene glycol transformation in yeast. It's a very generic protocol in yeast. Every yeast researcher uses it. So if you have a colleague that has some experience with yeast, also worthwhile just asking that person. And if you want to know a little bit more about the molecular mechanism behind homologous recombination, because I think it's really interesting, it's a very powerful tool. Also very important for the outcome of genome engineering, for CRISPR recombination, always very dependent on what mechanism of repair your host organism actually uses. I recommend you to start with a very nice review in annual reviews in genetics about kind of recombination proteins in yeast. And with this, I'm gonna stop sharing my screen, and we're open for questions.
Yeah, we have a couple questions. So I think we didn't answer this one yet, but does the matter or does the order matter when you use Gibson assembly? Well, that's one of the nice things about the one pot side of it. Once you've made your fragments and you've gone through all that process, it's just a matter of mixing them all together, and the Gibson master mix and all that and the homology will actually assemble it all together. Another question, can we make a plasmid which is a combination of a bacteria and yeast cloning sites?
So it depends on what you exactly mean by cloning sites. So usually, let's say DNA is DNA. So what you can assemble it in in yeast or by Gibson assembly. But of course, what you need to take into account that the genes or the DNA that you clone has a meaning, so it needs to be translated to a protein, and that needs to work via promoters, for example, and it also needs to be terminated by terminators. And these are, of course, host specific. So you cannot use a bacterial promoter and hope that it works in yeast. So that won't work. So let's say, you need to think about the functionality behind your DNA. So what we talked about today is really how do you, like the technical part of like, how do you assemble fragments of DNA together? But of course, the next layer is to think about what do they encode and where, like what I want to do with it afterwards. And then you need to decide, which host you actually work in.
And another one we have, to make E. coli accept an exogenous metabolic pathway, let's say 10 enzymes, would you use yeast assembly or something else?
Yeah, I think we discussed, so 10 enzymes are already a lot of components, especially given you might also add a promoter or a terminator. I mean, in some cases, you can actually use a natural promoter and terminator, and you can kind of like PCR that in one as one component, but 10 enzymes is a lot. And personally, having a lot of experience with Gibson assembly and yeast, I would probably go for yeast assembly. You can also consider sometimes what we talked about before, the functionality of the components. So if you're not sure that, for example, the 10 enzymatic pathway implies metabolic burden to your cells, you rather want to clone that in the non-native host. So for example, if you want to clone a 10 enzyme pathway with E. coli promoters, you could consider that you want to do that in yeast, because yeast can't really translate it, so it won't pose metabolic burden on yeast, so it won't be selected against. So likely the assembly works better in yeast because yeast doesn't actually know what to do with it. In E. coli, I would always be worried that maybe one of the enzymes is kind of like toxic, and then you won't achieve the full assembly. So this is kind of like, let's say, points of consideration you need to keep in mind when cloning.
Our fragments were constructed for Gibson assembly. In the case where it doesn't work with this technique, could these exact same fragments be used for yeast assembly, or do they need to be constructed differently, in a different way?
Okay. Can you repeat that question?
Sure. This one was in the main chat. So our fragments were constructed for Gibson assembly. In the case where Gibson assembly doesn't work with this, where it doesn't work, could these exact same fragments be used for yeast assembly, or is there something we need to do to change them?
Yeah, so basically, yes, you can use them. Again, the only thing you need to take into account is the backbone you clone them into. So and this only matters for the pieces that actually gonna do homologous recombination, or gonna be recombined with the ends of the plasmid. So likely those two ends need to be changed, but all the middle parts, they can be the same. Ideally, yeah, they can actually be the same. To answer that question briefly, just consider the backbone.
Cool. Is there specific criteria for choosing a particular yeast cloning cassette?
Again, this depends a little bit on the definition of yeast cloning cassette. Specific criteria. Maybe, so let's say if you talk about the yeast backbone, then there is not necessarily a specific criteria. So again, it depends if yeast is your eventual host you want to add your functionality to, or if yeast is only your cloning workhorse. If it is your eventual host, you of course want to take some things into consideration. For example, there's like high copy and low copy cloning vectors in yeast, so you know that might change the eventual expression level of your genes, so you might take that into consideration. Then you have different selection markers. So you might want to take into consideration how many plasmids you actually want to use in yeast. So actually you can use as many different in yeast as you want, unless you run out of cloning markers.
Mm-hmm.
So again, if you want to have 2 plasmids in yeast, you want to use different selection markers. But yeah, other than that, yeah, given this, the cloning cassette was referring to the actual backbone.
Yeah, it can be a bit different between different yeast. I know I spoke about Pombe, so commonly we'll use adenine, uracil, or leucine as our oxytrophic markers. And really the key thing there is you actually need to think a lot about your strains. So you need to make sure if you're going to do a double plasmid where you're going to have one that's adenine based and one that's leucine based, that you have a double knockout for both of those genes that you're actually transforming it into. Otherwise, it's probably not going to work very well. And promoters also can be another big thing. I don't think this one works in Cerevisiae, but in Pombe, no message thiamine is a common promoter that's used to actually drive expression in these plasmids. So, yeah.
I mean, in yeast, like, yeah, so Pombe, I'm more a Saccharomyces person. So we do have the most commonly used inducible promoter is the galactose promoter. So this one is repressed. It's, let's say, comparable to the arabinose glucose system in E. coli. This one just, you know, is induced by galactose and repressed by glucose. And we have a lot of kind of like endogenous promoters with different strengths. So there's a lot of work done by now on comparing promoters in yeast, which I think is also answering or referring to a question, to another question, which says, can the genes assembled in yeast be segregated and expressed separately? And yes, so like I want to mention, so in mammalians, not in mammalian cells, in eukaryotic cells, the genetic architecture doesn't work via operons. So I think that is something you find in prokaryotes. So this might be already the first component of your question. So in, you know, in genetic engineering in bacteria, you naturally think, oh, let's just like you have one promoter and then like have all these other genes, you know, be controlled together as operon. That you don't do that in eukaryotic cells. So in eukaryotic cells, you always have kind of like a transcriptional unit where you have a promoter, a gene, a terminator, promoter, gene, terminator. And as such, anyways, every gene is kind of like expressed by itself. And again, yeast assembly, depending on what your final host is, you can consider then yeast as just your cloning host or really the final host for functional expression. And then there's also actually a good question, how transformation efficiency, like how about transformation efficiency for large assemblies, yeast compared to bacteria? I can also answer that. So usually, I personally always get more colonies after yeast assembly. So I personally have never done a real kind of like Gibson versus yeast assembly experiment. So I can give you hard numbers here. But if you do a 6 to 8 piece assembly in yeast, you always have, let's say, at least 10 to the 2 colonies, like 100 to 1,000 colonies on your plate. While with Gibson assembly, I sometimes do not get any, or maybe only 2 or 3, while the overall efficiency of that batch is maybe, you know, 10 to the 7 per microgram of DNA. So usually the transformation efficiencies between yeast and E. coli, as expressed in, you know, colonies per microgram of DNA, I think are comparable. But then after the actual, you know, process of assembly, I usually get higher efficiencies with yeast assembly.
The one thing I would say in that regard, just from my experience, sometimes you'll express a gene that the yeast really doesn't like, and you'll get very, very few plasmids, or you'll notice that it'll start to grow very, very slowly. So that DCP1 that I'm using here, I know Sonja said that typically it takes 2 to 3 days to get colonies. That took me a week to get a colony because the cells just really didn't like them.
Yeah, which I think is also a good example of like, you know, which host you actually clone in, because maybe you could have cloned in E. coli and E. coli wouldn't have, you know, wouldn't have had the right promoter to express it, and then the actual assembly would have been easier in a non-endogenous host. Then we do, could you explain how the auxotrophic markers work in a little bit more detail? So yes, so in yeast, so in bacteria, we use basically antibiotics as selection markers, where you have a small molecule that you add to your plate, aka an antibiotic, let's say ampicillin, and then the bacteria become encoded on the plasmid a resistance gene that enables the one that encode for the plasmid to grow on that plate. So in yeast, we do not, meanwhile, we have some like, you know, real antibiotics, but what you could do also to select is just delete genes from biosynthetic pathways of amino acids. So let's say you have the methionine amino acid pathway, and then you delete one gene out of it that encodes for an early step in that pathway. So the yeast can't make methionine anymore. So then you grow them on media that, you know, lacks methionine, and of course the cells can't grow. But then you, on the plasmid encoded, give them back this gene, and then you select on media, on the same media lacking methionine. So you basically select via auxotrophic markers. So only cells that kind of like complement via the plasmid for the deletion in this biosynthetic pathway can grow on your plate. But of course, you need to keep in mind that the actual host cells need to be engineered to have these specific surgeries in their biosynthetic pathways.
You can do antibiotic selection. The main reason it's not done as commonly, or it's much more selective in yeast, is you have to use much more expensive antibiotics. So it's often cheaper and more costly, like just easier to use auxotrophy.
Yeah, and then we actually have an interesting question that goes back to the metabolic burden and the cloning. So for a large pathway, you mentioned that it could cause metabolic burden in some cases if done in E. coli. How would this issue be solved by cloning the pathway in yeast previously to inserting in E. coli? It wouldn't solve the problem of the metabolic burden, but it would enable you to clone the pathway. So in E. coli, you wouldn't maybe even be able to clone the pathway, and then you wouldn't have any data to show that it causes metabolic burden. In yeast, you would be able to assemble the full pathway, because yeast, you know, doesn't know how to express the genes from E. coli promoters. So you have the full pathway, you can bring it in E. coli, and then you could, for example, compare growth rates. So the main point here is not to say it reduces metabolic burden, it just enables you to actually clone the pathway. That's the distinguishing moment. Would adding a linker sequence between 2 genes, 30 to 50 base pairs, be recommended using yeast assembly or Gibson assembly fine? Yeah, I think I shortly alluded to that. So you can do that by Gibson assembly. In my experience, you just need to screen more colonies because you frequently find mutation in that regions, while you don't see that for yeast assembly. But, you know, you will be successful with both methods. It's just a matter of how many you have to screen.
I think we got them all for now.
Yes, I think so too. Yeah, I think if there is, no, maybe in the chat. No, I think the chat is also done. Great, a lot of questions.
Yeah.
Very great audience. Yeah, and yeah, if there are no other questions, I think we
I think we
call it a day.
got one more. These assemblies need long primers. Do they need to be purified after synthesis?
Yeah, that's also a good question. The answer is not necessarily. I never order purified primers. That doesn't say that it wouldn't hurt purifying them, but it's extra cost. So the answer is no, to keep it as cheap as possible. Great. Then, yeah, thanks again, guys, for being here, and we wish you a great summer. And as we said, you find the recorded webinar on the website of the Measurement Committee webinar page, together with all the other webinars. Great.
Thanks. Bye, everybody.